Okay, so we just have one little section left to finish up in our chapter on chemistry. It's a long chapter, so I know it's a lot of sections to cover, but we have it's 2 or 3 slides left. And so we're going to look at DNA first, specifically here. DNA, of course, is found in our cells, and primarily in the case of what we call eukaryotic cells, which are cells that have a nucleus and generally more complexity than prokaryotic cells have. It's found in the nucleus, at least in eukaryotic cells. The function of DNA primarily is to contain the recipe that it takes to build proteins. All right, so we're containing a recipe to build proteins. And so we talked about before, DNA is a polymer. It's made up of monomers, and those monomers are called nucleotides. Nucleotides then each are made up of a sugar, a phosphate, and a nitrogenous base. In DNA, we're talking about A, C, G, and T, as opposed to RNA, which will be a little bit different. RNA is very similar to DNA. There are some differences, though. RNA, first of all, functionally is a sort of a middleman molecule. That when you're going from DNA, which is nucleic acid, to protein, which is a very different kind of chemical language, RNA serves as kind of a middleman. So when it comes to ribosome structure to build the proteins, when it comes to carrying information from the nucleus to the ribosome, when it comes time for molecules to find the amino acids you need to build a protein inside of a cell, all those functions are carried out by RNA. Right, so RNA contains ribose instead of deoxyribose, hence the name ribonucleic acid versus deoxyribonucleic acid. RNA is ACG and U, whereas DNA is ACG and T. We talked about that before as well. And RNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. So we have those three main differences between DNA and RNA. The number of strands, the bases involved, and the sugar involved. Right? So we talked before about how DNA is a recipe to build proteins. If you look at the entire genome of an organism, that would be like the entire recipe book or the entire cookbook. So you take all the different recipes collectively, that makes up the cookbook. In the case of DNA, if you take all the genes collectively, that makes up the entire genome of an organism. So different genes then are sections of DNA that are used to build proteins. We have this idea of the central dogma in biology, whereas DNA is used to build RNA, and RNA is used to build proteins. And that flow of information is really what it comes down to in biology. That is the meaning of life. DNA is turned into RNA, and RNA is turned into protein. Now, there are names for those processes. Turning DNA into RNA is called transcription. Turning RNA into protein is called translation. We're going to get into those in much more detail in later chapters, but that is the basics of it. DNA to RNA, RNA to protein. That is the central dogma, the central main functional idea in biology. As far as some other nucleotides that we look at that are not involved with containing information about how to build proteins, ATP is one. ATP is also known as adenosine triphosphate. Notice it looks like a nucleotide. It has a sugar, it has a base, and it has not one, but 3 separate phosphate groups. And those 3 phosphate groups are important because they're a way to store up potential energy. When you create this molecule, when you take adenosine diphosphate with 2 phosphate groups and add a phosphate onto it, you make adenosine triphosphate. And that last bond between phosphates where you store up the energy that cells use for just about everything they do. That last phosphate to phosphate bond is a form of potential energy which can be used by cells to do different kinds of things. There are also 2 other molecules which are electron carriers. In chapters on cellular respiration and chapters on photosynthesis, we're going to see a need for molecules to carry electrons from one part of a cell to another. We do that with NAD+ and FAD. These are kind of like little electron shuttles inside of cells that are carried from place to place, from reaction to reaction, where they can be used to do different kinds of things. So we'll see how those are used later on in different chapters as well. There are some other coenzymes involved that are nucleotides too. We won't see too many of those in this chapter, but we may come across 1 or 2 of them as we go along. So that is the chapter on chemistry. Like I said, this last section was very short, but that wraps it up. And so from there, we'll move into the cell.